doula. So we had a home doula. And that's where she wanted to go. With the hurricanes, I think it was after Hurricane Ivan, the tree burned. And not the tree burned. And lived in the tree forever since. But she kept wanting to go home. So in the house that's they're all abandoned. But in the house, I went to the house and I got pieces of the house and I made a cross out of it and I blessed it for her. But she and then she was all
so
happy.
sweet.
So but yeah.
That was really thoughtful.
I love it. And then I made one for all of them.
For everybody. Let's see how
I
funny.
don't know who it was. The state or whatever, but they were just so beaten up and tear down. So, like Hurricane Rita, there was water all the way up to the attic, so that made it like bad. So we had to tear it down this past year. But
So we did that. Well, I
that's
can keep track of where we are. Here we go. I think. Yes. Okay. Minimize this. We have iClicker attendance. Log in.
they use some alarms. There's like these cheap alarms that you can put to open up the door. No, the family doesn't care.
Okay. Get our attendance going. All right, so attendance is up if y'all want to go ahead and do that. And then I want to pull up our Moodle site real quick for here we go. Final exam. Okay.
Each different section, what we created.
Good morning. Don't forget a wipe. I care about y'all. Don't forget a wipe. I'm not even on the screen for y'all. I'm worried all about the online people. It'd be nice to show the screen for the people. They do outnumber y'all today. They do. They indeed do. If the Wi-Fi goes out, I don't even want to think about it. I don't even want to think about it.
If the Wi-Fi goes out while I'm making an online final, I'm just
Oh, no. You know, so we're going to talk about the final.
yeah, okay, so there's too many people to be in this room, right?
We're not, so I'm going to talk about it.
Okay.
Okay, so it's 10:45, so I'm going to go ahead and talk about it. Now that class has started, so make sure you check in on attendance because that's rolling right now. But I want to talk first real quick about the exam. So our final exam, remember it is semester cumulative. So what chapters are those? 1 through 11. Okay, our final exam will be in person, on paper. On paper, isn't that awesome? On paper. Wednesday, November 18th, from 1 to 3 p.m. in, look in bold, Pelte Auditorium. We cannot all fit in this room. So I had to find, goodness, it was crazy to try and find a room, but we got it. So they're really small desks. They're the little ones, but it's the best I could do for us. But I thought it might be nice not to have to worry about the internet crashing on you during your final exam. Let me pull up my chat because I don't think my chat's up. Okay. So, questions about that? And it will be 100 points like exams 1 through 4. It's not more heavily weighted than anything else. Okay. I'm still working on it right now, so I don't know how many questions it will be just yet. I'm still, it's in progress. But it is not going to be a duplicate of your exams 1 through 4. Yes, you should look over your exams. I I absolutely would suggest going over your old exams. What did you get wrong? What did you get right? They're good refreshers.
Study the PowerPoint and try to repeat it.
Yeah, you could study the PowerPoints. You could try to retake the exams. Those are good ideas. But the more specific questions will be for chapter 11 because you haven't been tested on that yet, right? So you can definitely expect to see a discussion question on chapter 11. You know, one of what?
Will there be more of chapter 11?
There will be more of chapter 11 than any other individual chapter, but the majority of the exam will not be chapter 11. Does that make sense? Like, yes, chapter 11 will be more weighted than the others because you haven't been tested on it yet, but not, it won't be like a chapter 11 test with a couple of review questions. Yeah, okay. No, it will be more questions. So Logan asked, will it be about the same amount of questions? No, it will be more questions. There will be more questions because it's basically double the time, right? So it won't quite be double, but it will be more. It will be more. Okay.
Same amount of discussion questions.
The discussion questions will cover one sheet. Right? So you'll have all multiple choice and it'll not be a Scantron. You do not need to bring your own Scantron. I will have, it's similar to a Scantron. It's called a zip grade sheet. Annie knows, a few students who've had me in the past know. But it's like a Scantron, but I'll supply it. Okay? I'll supply that sheet. I'll supply your exam on paper. I'll supply on paper the exam resources that you had on the last exam. So everything, the codon chart, the periodic table, the glycolysis, the citrate cycle will all be there. Okay. Trying to think what else. So that'll be multiple choice, and then the back page will be discussion. But it'll just be one page. Maybe 3 questions, something like that. You have to have room to write it. Like the questions and your answers will all fit on one paper. No, it won't be 20 questions and then you have to lose sleep. No, everything I plan to grade one sheet for every individual student. I do not have time to grade 20 free response questions from everyone.
We only have to choose one.
No, it won't be like that. No.
Not that.
The thing is, you'll have a few questions on the back page of your exam. A few questions. The questions will be here, and your responses can be written on the same one sheet of paper. Okay. Any other exam questions? Trying to cover everything, and I will remind you again on Friday that it's in Pelche. I'm going to send you an email the night before that says it's in Pelche. You know, so make sure you go to Pelche. Other questions? Good on that? Okay. So the only other thing that's going to be graded between now and the end of the semester is your chapter 11 self-check. Okay.
And our little lab report. In our lab report.
Well, that's lab. That's lab. So yeah. All right.
I'm sorry.
No.
For lab, do y'all have like a certain amount of like questions or anything like y'all start the exam?
Yes. So the exam, so the question is about lab exam. Lab exam is going to be questions that you have to do similar calculations as to what you've done in the lab. Some questions on the theory behind the experiments that we do, and some questions on like how the experiment works.
So like the procedures.
Sure. So question was procedures, yeah. But it's not meant to be an exam that like catches you or anything. You know, this is meant to be an exam that if you've done the labs, you're fine.
Right. Just to review it to make sure that
Yep. So make sure you have whatever you think you might need in your lab notebook, because it's open lab notebook. right? So if you showed your work for your calculations, you're gonna be fine. You know what I mean? Okay. Any lab questions to do with the additional problem sets? If there was an additional problem set that was assigned for a lab, yes.
So, it's the lab, but it's the additional problem sets right after the chart of the way you put in your absorbance is what I was.
Well, it's the additional problem set.
He's in my group, and Dr. Smith didn't say anything about an Excel, didn't say anything about additional problem sets like this.
Well, you're not gonna have to do anything in Excel. I mean, you for the lab report, yes, not for the
Right.
exam.
No, for the lab report.
Oh,
Like, I don't think he's asking about the additional problem sets for this past lab.
I don't think we've done additional problem sets for like the last.
no, they're in my, so if you were in my section, I told you which things I wanted you to answer. At the top of that printed out sheet of paper, I told you which ones to answer.
Yeah, I think we only had to do that like maybe first two.
No,
Yeah.
he said it's the last one questions after.
Like,
Oh, for the last lab, there are a couple of questions after the chart. Yes,
not like
yes.
the additional.
Not the additional problems, no, but like the questions. Yes. Okay. William, I can't answer for Dr. Smith, but I can tell you that I'm not going to ask you what were the brand names and item numbers for reagents that you used in lab.
Oh, yeah.
That is not going to be on the exam. I sent out an email this morning that lab final is going to be Thursday from 6 p.m. to 10 p.m. It is open for a 2-hour window. It's open for a 4-hour window, but you only have 2 hours to take it in. Does that make sense? Yes, you will get all of your past lab reports back before the exam. Absolutely. I have them almost all graded. I'm so close. I'm going to hand everything back to those who I see on Friday. And then if you want to come and pick up if you're in my A group and you want to come pick them up from my office, you can. I'll send out an email when they're all done. All right. Taylor, would you know if our last lab reports are due this week instead of next week? I don't know, Dr. Smith.
He said for the last, like the very, he's asking about the.
That's what they're asking at the very last
He
lab.
wants it before the final exam, so like Wednesday is your last.
Okay.
day,
Okay. So the response is that he wants it before your final exam.
so Wednesday.
So if the exam's on Thursday, he wants it on Wednesday.
Just, that's the last day, he would put it Monday, Tuesday, the last, the very last.
On Wednesday. So for me, I was gonna say on Thursday, but he wants it on Wednesday,
Oh, no, like he's just saying before the final, so I guess.
before the final.
Yeah. just-
Thursday as of yesterday. Okay, and me too. That's the same thing for me too. The Thursday before the final. Okay,
I don't think you'd be too specific about this, as long as you can.
and I have a folder
Yeah.
file on my door
Very, very
that if I'm
well.
not in my office, you can just stick it in my
Yes,
folder.
it's due next, yes.
Okay, does he want it emailed to him or on Moodle?
No, he doesn't want to email to him, he wants it personal in hand, so he said if he's not in his office to either put it in his mailbox or sweep it under his door.
Okay, so he said that he wants it in person, to not email it to him and not to put it on Moodle. So you can either slide it under his door or you can put it in his mailbox in the main office. One of those two things. I will take it on Moodle, or you can put it on the folder on my door. I will take either. So the final is next Thursday. Taylor, the lab final is next Thursday from 6 to 10 p.m. Okay, okay. All right, so did I answer everything? I don't even know. There's so many questions.
Yeah.
Did I answer? Okay, just repost your question if I did not answer your question. And I will try to answer it again. And then I have some good news to tell you. You said we have 2 hours. Yes. So like the timer will start for you to take your exam and you will have 2 hours to take it, but the exam will be open for between 6 and 10 p.m. Will final consist of new questions? For the lecture final, it will be new questions. Colin, new questions. Somebody else had a question in class. Yes.
If we have like any trouble, because like in the past, we've never got any response from that, just like an email, and I don't ever use response, like I don't know if anything was to happen, a glitch or whatever. But if we were to have any issues, and like my plan is to do 6 o'clock, so that making sure there's nothing wrong. Is it okay if we can email you if we cannot get in touch with Dr. Smith?
You can email me, but the thing is, so the question is, if you have trouble during your online lab final exam, and you can't get in touch with Dr. Smith, you can email me, but I can't do anything because I don't have access to you in lab on Moodle. So I can't do anything.
I gotta know if like you can contact them.
I would say, what I would say is, I would suggest go to his office and say, okay, what happens if we have trouble on the
He
during
says
the final
that
exam?
to email me, but we don't get a response back until hours.
You can email me if you're really struggling. You can email me, and I can try, but I can't make any promises.
No, that's what
I can
we're
try.
scared about is like if something was to
Happen.
and
Yeah,
run on.
Sure, you'll have his email.
I'm sure he'll have his.
He's
Okay.
still very scary about it.
We
I don't
know
think
it.
it's gonna be a problem.
Yeah, I don't think so either.
He's very
Okay.
relaxed, so I don't get
All right, so let's see. Will the final exam, Colin, answer that? Yes, Austin, he wants to see. Okay, so that was not for me. All right, are we saying, no, it'll be a similar exam because we're testing on the same topic, but it will not be exactly the same exam. When you said Thursday before the final, did you mean? No. I mean before 6 p.m. next week when you actually have your exam. That's when your last lab report is due. Emily, does that make sense? Yes. Okay. Okay. Any other questions? we had to get through a bunch of stuff. All right. So y'all know that so do we have to arrive at 6? You can start the exam at 9 p.m. if you want. I would not suggest it. I would highly not suggest it. And if you're going to take a respondus exam and you're not here in the classroom with me, right? I won't be with you. I would try to take it on a computer that is hardwired to the internet so that you don't have internet issues. You know, that's my other. So there are lots of computer labs on campus. If you look on my website, on my Moodle, you can't come here. You won't, the doors will be locked. But here are places, look here, locations on campus that have computers with respondus, right? You could do it at any of these locations.
And they don't close at a certain.
And they don't close. They will be open. Okay. All right. So you can arrive at 8. You can arrive at whatever time you want, but it will close at 10 p.m. And the maximum amount of time that you will have with the exam open is 2 hours. But it shouldn't take you 2 hours. It shouldn't take you 2 hours.
It's gonna be the same format as like the lecture.
The multi-purpose room in the library closes during finals week. I think during finals week it stays open. I can double check. I can call and check. Okay. All right. Any other questions? No? Okay. So I have one last thing to talk about. Y'all know when you open Moodle and you get that little pop-up that says you need to evaluate your classes? Okay, so right now we are sitting at 38% of the class has evaluated the course. I will make a deal with you. If we can get 80% or more to evaluate the course, I don't see what you say. I just see what percent has done it. That's the only thing I can see right now. I will give you 5 bonus points on the exam.
Get on your ass and do it.
Please do it. 5 points on the exam. Okay, 80%. So I don't know who does and who doesn't. I just know that 80%.
Don't you? This is
When are
not
you certain time?
Don't you?
Opens up the survey. Everybody go take the survey. Be honest in the survey. Give me as much feedback as I can get to help make this course better. Tell me what's good. Tell me what you would like to change. Give me everything you got, okay? All right.
At a certain time, you want 80% or?
I think it closes on Friday.
Okay, so.
I think the last time that you can enter it is Friday. So just before Friday, 80%, and everyone across the board gets 5 points on the final exam.
I don't know if you said this.
Ask-
Like how many questions?
Are going to be on the final. I'm not sure yet, because I'm trying to balance the discussion with, you know, how many multiple choice. So I'm trying to make sure it's not too long. So I don't have that answer yet. It's a work in progress. Okay. Okay. Now I have one other piece of news. Did y'all hear in the news that there was a guy who ingested the alpha L glucose form? Did y'all hear this? They actually found out that he didn't have any ill side effects because he's ambidextrose.
Oh, yeah, I
know. He's ambidextrose.
Yeah.
Ah. No, there wasn't. Nobody really did that. You would really, you couldn't digest it. So if that was your only source of energy, you wouldn't survive. But if you eat some of it, your body just can't metabolize it, and you would pass it. Right, it would come out as waste. But I thought it was funny. Okay, all right. We actually have to do some science today. All right. So if we don't finish all of chapter 11, it's okay. We're just going to do what we can and roll with it. Okay. Minimize all of this kind of stuff. Okay. Here we go. All right. So now we're going to talk about details of the electron transport chain. No matter how many times I see system, I just can't do it. Right? All right. So where does oxidation of NADH occur?
In the mitochondria.
Where in the mitochondria? On the what? True in complex 1, but I'm thinking like of all the different locations in the mitochondria, the outer membrane, the inner membrane, the matrix. What? So it's on the matrix side. Matrix side of what? Which membrane? There's two, outer or inner? Inner. Mitochondrial membrane. How many electrons does NADH donate? 2 electrons. Okay, so it's going to go to complex 1. Remind me, what was the name of complex 1? It's the NADH ubiquinone oxio reductase. All right, good, good. Got to get comfortable with those names, right? Okay, so our pair of electrons are going to enter through NADH, and then where else do we have a pair of electrons that are going to come in on this? There's one other place as well. From FADH2 oxidation, right? So how many electrons are going to come in through FADH2? 2 electrons. What complex are they going to come in at? They're going to come in, well, not the third. What oxidizes FADH2? The second, complex 2. And complex 2 is in the citrate cycle, which is succinate dehydrogenase. Okay. All right. So we're going to go through multiple redox reactions to make this work, right? you are actually going to go through a minimum of 20 steps in order to make this happen. Now, you're not going to have to know the name of every single part, right? We're just kind of naming the big parts. But once you get to those complexes, once it's in the complex, it's going to make multiple jumps inside the complex, those electrons. And so you're not going to have to name every jump, but you do need to know the complexes. And at the end, we're going to reduce our gaseous oxygen into water, right? So O2 will be reduced to water. So how do we make those 20 jumps? What's the requirement? How do we have this a sequential arrangement of electron carriers? With the reduction, so it's exactly right with the reduction potential, increasing reduction potential. So it's due to increasing reduction potential. So let's kind of look real quick at a diagram of overall what's going on. So what's the difference between A and B? Take a look on your notes. What's the difference between A and B?
B doesn't have that big, B doesn't have that big subunit in it.
What does that mean? What's the conversion of NADH? Well, so the first one is what is NADH? Why did I do that? The first one is NADH, right? Do you see that? We're starting with NADH and we're going to reduce it to NAD plus. And here's our path of electrons. This is where they're going, right? What's the difference between A and B in terms of that? Are we starting with NADH and B? No, we're starting with FADH2. So it's the same thing. They're both the electron transport system, but we're showing, okay, what happens? Where does NADH come in? That's A. A, we're tracking just NADH. B, we're tracking FADH2. So NADH2, I'm sorry, NADH2. NADH is going to come in at complex 1, right? Complex 1, we're going to exchange NADH for NAD+. We're going to pick up 2 electrons. And that's in the NADH ubiquinone oxioreductase. Now, when those electrons are eventually transferred to quinone, right? That's the little Q. That's this guy right here. Once we get those electrons transferred, because it's going to go through a couple of steps, what is changed in this system? We're going to pump 4 hydrogens. So this is how we write it. 4H plus are going to go from the negative side to 4H plus going to the positive side of the membrane. This is our notation. So you do it as like a subscript. Q is going to become oxidized into QH2. Right? Because in addition to carrying those 2 electrons, right, we're also going to pick up 2 hydrogen ions from the mitochondrial matrix. So now we're at QH2. QH2 goes to complex what? Complex 3. We're going to skip complex 2. We're going to complex 3. Complex 3 is our ubiquinone cytochrome C oxioreductase, right? So those 2 electrons are going to get shuttled around, and they're eventually going to get picked up one at a time by cytochrome C. When we get to that point, what job did we accomplish? We picked up 2 more hydrogen ions, plus the hydrogen ions that were attached to Q, and we're going to pump those out. So what number are we at now? Now we have another 4 hydrogen ions that are going from the negative to positive. Okay, so how many hydrogens have we pumped so far? 4 of 4, 8. We've done 4 in complex 1. We've done 4 in complex 3. So we have a total of 8. All right, 8 hydrogen ions so far. Now, cytochrome C gets those electrons, right? Takes it to complex, what? Complex 4, which is the cytochrome C oxidase. Now those electrons get shuttled around, eventually coming back to reduce oxygen to water. In that process, we're going to harness that energy and we're going to pump 2 more hydrogen ions. So 2 more hydrogen ions go from the negative side to what? To the positive side. Okay, so how many total hydrogen ions have we pumped? Total is 10. Okay. Now, what's up with FADH2? FADH2 is going to donate its 2 electrons, right? And it is going to participate in reduction of this QH2 in order to get those 4 hydrogen ions out. And so you're not getting additional hydrogen ions, but these electrons that come from FADH2, whether that's from the citrate cycle or the fatty acid oxidation, you're going to need FADH2 to complete this cycle. And I'm going to show you why that is today.
So why did you say
We skipped step 2 because the electrons that come from NADH2 don't go through complex 2. I'm sorry, the electrons that come from NADH, why am I putting a 2 on it today? I do not know. So the electrons that come from NADH bypass complex 2. But in order for complex 3 to work, you need the 2 electrons from NADH and you need the 2 electrons that come from FADH2.
So it's not that you're not using complex 2 in the thing. It's just the 2 doesn't go through.
Correct.
So
Correct.
So we bypass the Q's.
The electrons coming from NADH bypass complex 2. But in order to make complex 3 work, we need the electrons from NADH and we need the electrons from FADH2. Okay? All right. So if at any point you inhibit any one of these steps, you are going to inhibit the electron transport system. So y'all know, like you've probably seen in the movies, right? The Amazon tribes that would have those poison darts. Yeah, so that comes from a root, a plant root that produces this to inhibit like bacterial growth and things like that. And that's this inhibitor right here. We talked about hydrogen cyanide in gas chambers. Carbon monoxide will have an effect on the electron transport system. Antimycin A is one that it's an antibiotic that's produced by some organisms, right? But if you inhibit any one of these things, and it's any of our complexes, if you inhibit the ATP synthase complex, if you inhibit the pumps, that's not the pumps, but the channels that allow ATP and ADP in and out, then you're going to have inhibition of the electron transport chain and cell death, right? So any point in this cycle, you can inhibit everything. All right, so how does all this, yes, sir?
Can you go back to the 2 figures?
Uh-huh.
Okay, for figure A and B, are they combined together, or is it only figure A that gets you the 10 hydrogen ions?
In order to get the 10 hydrogen ions, you need a combination of A and B. And I know it's a little confusing right now, and so we're going to go through it step by step, but I want you to know on the front end that you're going to need both NADH and FADH2. So
You just the fact that it's separated them, it makes it seem like it's 2 different things.
yeah, it has to do with the quinone, the ubiquinone, but it's a big cycle, and so we're going to talk about it in detail, I promise. Yes. Uh-huh.
So, like, if you have an inhibitor on complex 4 and say, like,
An electron transport chain up to, so the question is, if you had an inhibitor for complex 4,
Yeah.
you would go through the entire the entire electron transport chain until 4.
You're putting more energy into, so like, would that be more cell death if you went underwent all the way?
It's very, very brief. It doesn't matter which complex you inhibit, cell death is going to be very rapid because you're not making ATP.
So it isn't like the same towards like how much energy you're putting into an
Mm-mm. No. Okay, so remember how we said what you need to do is change your redox potential, right? In order to make sure that those electrons change. So look at the numbers. What's happening to our reduction potential? It's generally increasing. Is it perfectly increasing? No, but what value is this that we're looking at? This is standard conditions. So tell me about standard conditions. Are they at cellular temperature? No, right? So the temperature is going to be different. How about normal cellular concentrations of substrates and reactants? No. So that's probably the #1 thing that's going to change and make this a perfect gradient of potential so that the electrons will jump is really concentration.
The potential, you say the potential increasing.
It increases. So look at here, the direction of electron flow. What's happening? You're going from something very negative, not very negative, but negative, into something that is positive, right?
Is that I think we looked at it in 10.1, the next equation.
Yeah.
That's what relates to the standard and the.
I'm going to show you. I'm going to show you. Absolutely, absolutely. All right. So how do we make this happen? How are we getting electrons moving just because of, how do we get protons moving just because of electrons, right? So how do we get those protons into the intermembrane space? Well, the short answer is we don't have all the answers. We're not 100% sure. This is an area of active research. We think there are 2 different mechanisms that are working depending on what complex that you're in. Over here, this is what we think happens in complex 3, a redox loop. So in a redox loop, basically you separate your protons and your electrons so that they're going on opposite sides of the membrane, and you go through what's called the Q cycle. And we're going to go into lots of detail about the Q cycle in just a few minutes. The other one is a proton pump. And a proton pump is different because in this case, when you pick up those electrons and you drop off the hydrogens, right? So it's both a change in electrons and a change in hydrogens. You're going to actually induce a conformational change in the protein. And when that conformational change occurs, now we can pump those hydrogen ions out or in, depending on what, whatever direction you're thinking. But we're going to pump them to the intermembrane space. So this is probably what y'all are more familiar and comfortable with, a conformational change allowing for something to happen, right? The Q cycle is pretty different, and so we're going to talk about this one in quite a bit of detail so that you can kind of understand it. So the proton pump is for 1 and 4. So normally, right, when you have those electron carriers, they're carrying the electrons and they're carrying hydrogen, right? And so in the Q cycle, in a redox loop, you're basically going to send off the hydrogens in one direction and send off the electrons in a different direction, instead of them going with the same molecule like you're pretty much used to. And so that separation of the electrons and the protons, you're able to shuttle the hydrogen ions out into the intermembrane space and then shuffle those electrons along a pathway. And so we'll talk about how that works in detail. I know it's not something that you're familiar with. So what we're going to do is we're going to go through the complexes. And when we get to the Q cycle, we'll talk about the Q cycle. All right. So let's start at complex 1. So at complex 1, what are we starting with? NADH, right? Okay. So this is really the largest complex, right? That was that L-looking thing that's embedded in the membrane. Yeah, it kind of looks like this, right? It's huge. It's like 15 to 40 polypeptides that are all hooked together. It's huge. And it also has a flavin mononucleotide bound to it, and it's the flavin that accepts the 2 electrons from NADH, right? Because you got to think in these complexes, we have to have things that are able to pick up the electrons. Remember we said it's going to take at least 20 steps along the way to get there. This is step 1. Okay, so FMN is very, very similar in structure to FAD. Very similar. And I'll show you a structure in just a second. Okay, so here's our schematic of just complex 1, right? We have NADH. NADH is going to give up its 2 electrons, and we're going to have an extra proton, right? Okay, so FMN is going to pick up those 2 electrons. Now, FMN is going to transfer those electrons to other electron carriers. And you see these electron carriers are abbreviated FES. What is that? FES.
Iron, sulfur.
Iron and sulfur, right? And so it's an iron that's stuck in between sulfurs, right? And so it creates a complex where you can reduce that iron and you can change the charge on the iron, right? So you can go from a 2+ to a 3+, and then you can regenerate back to a 2+. So this allows us to pick up those electrons and to just pass them along in the inside of the complex. Kind of like what? Kind of like hemoglobin and oxygen. Very similar, right? Okay, so we're going to pass them along, we pass them along, we pass them along. Do you need to know the individual names of these? No, you do not, but you need to know that they get passed along. Okay, now as that is happening, right, we're going to start pumping our hydrogen ions from one side, from the negative side to the positive side. Okay, when we get to the end, here's our next electron carrier that's not part of complex 1. This is a mobile electron carrier. Complex 1 is stuck in the membrane. Our ubiquinone is mobile. It can go and shuttle back and forth. Okay, so you have to remember that this one can move. So when ubiquinone picks up the electrons and the hydrogen, right, we're going to form a new complex, just like we did with NADH going to NAD+, right? So instead of regenerating from NAD+ to NADH, we're going to go from Q to QH2. All right. So now we have that first transfer, and we're going to have 4 protons that move into that intermembrane space. Now, this QH2 is going to complex 3. It is not going to complex 2. It is going to complex 3. All right. So if we look real briefly, you don't have to know in detail, but our flavin mononucleotide, right? Very, very similar in structure with our rings, right? And so it has nitrogens, and those nitrogens are going to pick up the electrons. So this one picks up 1 electron and 1 hydrogen. Then we go through a second reaction and we pick up a second hydrogen and a second electron. And so we get our reduced form of FMNH2. So it's very similar. It's not, I know it's like a new structure, but it's not something you've never seen before, right? Okay, so now we're at coenzyme Q. Coenzyme Q, we said, is mobile. It can move. And it's going to take those electrons from complex 1 to complex 3. So before we talk about complex 3, what was the result of complex 1? Let's back up just a minute. Result of complex 1. What did we get out of complex 1? See if I can do this. Okay, here we go. So what do we get? 4 hydrogens. 4 hydrogens going from the negative space to the positive, which this is matrix, and this is inner membrane space, right? Okay, what else did we do? Yes, you get QH2. So you have quinone, ubiquinone, which is Q, that is reduced to QH2. So ubiquinone is also called CoQ. Have you know about CoQ10 supplements? Have y'all heard that before? Some people have. Yeah, and it's just because the human version has 10 units of a side chain, and so they call it CoQ10. Other organisms have other numbers of chains, but you can buy that at the store. It's not regulated by the FDA, but it is a purchasable vitamin supplement, right? Okay, so what else? What else did we do? That's really important. We regenerated NAD plus, right? We went from NADH to NAD plus. Okay, so if we wanted to write an equation for what happened at complex 1, what would we say?
-4H plus N. Plus N.
Well, now be careful. Be careful. It's not 4. It's not 4 because we required, let me go back. Let me go back to our picture, right? What happened here?
6.
We had to use up a hydrogen here in order to,
Okay, so 6.
well, actually it's actually only 1 hydrogen, and this one right here. It's only 1 hydrogen per NADH molecule. It's only 1 in the net reaction. Okay, so we're actually going to make, ooh, I went too far. Did it save my work? Where did I put this? Here. I'm losing it. Okay, plus 5H plus. Okay, now what? We need something else. Q is going to turn into what? QH2 QH2 plus NAD plus hydrogens. We have a hydrogen here. So it's 6 hydrogens on this side. It's a total of 6 hydrogens on this side. So we need 4. And so these are on the negative side. These are going to be on the positive side. The other hydrogen is coming from here. So we have 6 total. 5, 6. So 2 and 4 is 6. The 4 here, it results in a total of 4 being pumped into the inner membrane space. It's NAD because it's NADH plus H plus. Really. So we just took that here and we left it here, but it's going to stay in the matrix. It's not being pumped out. There are only 4 that are being pumped into the matrix. But 5 are being used in the reaction.
Right. But
Yes.
5 is in the matrix.
Yes.
Okay.
Plus this one that's also in the matrix because it's attached to NADH. Right? Okay. All right. Yes.
You come back to the previous slide on complex 1.
Complex 1. This one? This one.
Okay, so is FMN similar to FAD or FMNH2?
It's similar to FAD plus because it's going to pick up, right? FAD is going to go to FADH2, right? So FMN goes to FMNH2. That answer? Yeah.
Okay, so it's similar process structure.
Right, it's similar in process because it takes them one at a time. And you make that weird intermediate. Yeah. Mhm. The extra hydrogen stays in the matrix. The extra hydrogen stays on the matrix side. Okay, so where are we? Whoa, too far. Okay, where were we? Here. All right. So coenzyme Q, right? This is the one that has our electrons. It's going to go from complex 1 to complex 3. We need those 2 electrons that are in the citrate cycle or fatty acid oxidation, right? That come from what other molecule? Right? This is our entry point from citrate cycle, from fatty acid oxidation. So we're going to have 2 electrons. We need 2 electrons from where? NADH. And we need 2 electrons from FADH2, right? We need all of those electrons, okay? So when we're converting our electrons, we're going to have to convert them from a 2 electron transport system to a 1 electron transport system, because coenzyme Q, or ubiquinone, carries electrons 2 at a time. Cytochrome C carries them 1 at a time. That's a problem. We need to be able to split up those electrons. Otherwise, our electron transport chain is not going to work. I'm telling you, this is highly, highly conserved across many different species. So we have to have a way that's going to make that happen. So how are we going to make that happen? All right. We have to talk about ubiquinone because it's called the Q cycle for a reason. This is how we're going to split those electrons up and how we're going to pump some more hydrogen ions. So we know ubiquinone is reduced to QH2, right? So here's ubiquinone, and this is the human one because it's telling us that we have 10 units of this hydrocarbon tail, right? So I can tell that this is the human ubiquinone. Other ones will have different numbers right there, and that tells you basically which organism it comes from. So what's happening with ubiquinone? We're going to pick up electrons and we're going to pick up what? Hydrogens. So we're going to pick up first 1 electron. And so what happens to these double bonds on the oxygen? They break, right? Okay. Now we pick up a 2nd electron and we pick up 2 hydrogens. So those hydrogens are each attached to where those oxygen double bonds were. So now we have our reduced form of ubiquinone, which is ubiquinol. Whew. I just write it QH2. Just write it QH2. It just makes more sense, right? It's easier. It's easier to understand. And so this is where, at that point where we have those first 4 hydrogen ions being translocated from the matrix to the intermembrane space. But we got to do that multiple times, right? So how do we get the additional electrons from FADH2 in order to be able to use them? Well, complex 2 is succinate dehydrogenase, right? Succinate dehydrogenase, we know from the citrate cycle, catalyzes our oxidation from succinate to fumarate, right? This is something we learned last chapter. So when we do that, we know that that converts FAD to FADH2. Now we have those 2 electrons that can be transferred to the iron sulfur complexes that are also electron carriers within complex 2. Once we do that, now we can also transfer electrons to Q. So we're going to reduce our coenzyme Q to QH2, right?
So this is a new Q.
This is a new Q. This is a new Q. So now we've trans.
This is all
A new Q.
This is a new complex. This is a new Q, right? This is a fresh Q. Now, where are we sending our QH2s? What's the next stop? Now we can go to complex 3.
I thought we were on complex 3 already.
Nope, we were not. We were on, this is complex 2.
Okay.
This is complex 2. Okay, so now we can move on to complex 3. The name of complex 3 is ubiquinone cytochrome C oxoreductase. So our point here is to move those electrons from ubiquinone to cytochrome C using oxidation reduction reactions. Love the name. All right. So what we're going to do is we're going to reduce cytochrome C, and when we reduce cytochrome C, we're going to translocate our second set of 4 hydrogen ions from the matrix to the intermembrane space. So we have docking sites on complex 3 for QH2, right? So on one side, it's going to dock QH2. On the other side, it's going to dock cytochrome C. Now, here's the little bit of a tricky part. There are 2 binding sites for ubiquinone. So it contains 2 binding sites for ubiquinone. One binding site is called QP. One binding site is called QN. Where do you think QP is? Positive side. Where do you think QN is? The negative side, and really the negative side is the matrix, the matrix. Okay. So we're going to have a similar transfer of electrons through iron sulfur clusters, just like we saw in the other complexes. And we have 11 different protein subunits that are going to be used to make this happen, because it gets kind of complicated. So if we look at our complex, right? Here's our complex. We got QH2 coming in. QH2 is going to bind in that first docking site. So you see how it's labeled QP, right? Here is QH2 docked in QP. All right. So we need, the goal here is to convert our 2 electron transport process into 2 1 electron transport systems, right? So our electrons come from that Q, from coenzyme Q. So the way this is going to work, or did I give y'all a? No, I didn't give y'all a more detailed. All right. So you're going to have oxidation of QH2, right? So we're going to convert QH2 back to Q. When we do that, let's extend the page. So when we oxidize QH2, what's going to happen to that QH2? Do you see how 2 electrons come off, but then get split? Oh, maybe. Okay. It might be that it's because it's 11:40. I don't know. That's weird. Okay. I'm unmuted. but it is 11:40. So we're gonna pick up right here where we left off. And whatever we get through on section 3, so on chapter 11 is what we get through. Okay. So we're probably not going to get to regulation, but I really want you to understand how the electron transport chain works.
Okay.
So whatever we cover is what we cover.
Staying at the chapter, basically, we're going to be staying at the section 3. Chapter 11.
Hopefully we'll get to section 3 through the end of section 3. That's my goal.
This looks like the self-check.
Yeah, it might. It might. Okay. Are you going to be posting videos? No, because whatever we get to in class is what we get to. You won't have any more videos to watch between now and the exam. What we get to is what we get to. Okay. But read through at least what we're going to cover. So read through at least section 3. Okay. Any other questions? Y'all, I was just in the zone. Yeah. Oh, hold on, Tristan. Hang on. Okay, Tristan, go ahead.
I'm confused about complex 1.
See, it doesn't. Hold on, wait. Start again. Start again. I'm sorry.
I'm just a little bit confused about complex 1, where all the hydrogens and the net reaction and the electrons are going.
Where are we? Here.
Right. So it's 6 electrons or 6 hydrogens that we start with in the net reaction.
No. So no. Here's 4 that are going to move, right? And here's our 5th hydrogen, and then here's our 6th hydrogen.
Okay. So we wouldn't count in the net reaction those 2 hydrogens over there?
No, because they're not actually leaving. They're not going anywhere. They're not being used. They're not participating in something. It's a net reaction, not a complete reaction. So when you're talking about net, you're only writing the things that will change, that are affected.
So they're kind of just, I guess, catalysts.
They're required for the reaction, yes. But you don't see a change in them in terms of location, in terms of what they're attached to.
So which 2 hydrogens are going to Q, to ubiquinone?
These.
Those 2. And that other hydrogen that's just sitting in the matrix, that's from
These?
the, no. Well, I guess they're both from the citrate cycle. So they both of those hydrogens go in with each of their electron, their 1 electron, and that's how that works, right?
Well, so no, the electrons are going through the chain here. Then you're going to take these 2 hydrogens and you've got 2 hydrogens.
Oh, so those are the same ones.
Yeah.
I thought since there are no free electrons that like the hydrogens are going with them.
Well, you got to think that there are hydrogens floating here. So they might not be the same hydrogens. They might not be the same protons, right? But you're putting 2 in and getting 2 out.
Okay.
Remember we talked about the proton cycle? Like they're going to go into the matrix and they're coming back. They go into the matrix, they go into the intermembrane space, they come back to the matrix. Go into the intermembrane space, go into the matrix, right? So you always have hydrogen ions in both places, but you are going to have more in the intermembrane space, which makes it the positive area, and you're going to have less in the matrix space, which makes it the negative area.
Yeah, okay, thank you.
Good. Anybody else?
I was confused when you said 6 hydrogens, like 6 hydrogens overall, but
Yeah.
are they all in the matrix?
Are they all? They all start in the matrix.
So, but then just 4 of them.
Just 4 of them are pumped out into the intermembrane space. Yeah, it's kind of hard to keep track of the hydrogen ions. It is. It's a challenge. All right. Okay, so Taylor, did we answer you? Mhm. Yep, and these two hydrogens are then gonna make QH2. Yep, yep, good. Okay, anybody else?
I have a question, Dr. B.
Sure.
So with the NADH, where does the 2 electrons come from? Is that from the bond between the hydrogen and the NADH?
Yes, because
Okay.
this is going to be just a proton, right?
Right. So then whenever you go to FADH2, is the 2 electrons come from just the first bond of the hydrogen? Because it has 2.
Here, this one?
Yeah, so why doesn't 4 electrons come out of that?
Because you're not, let's see, how do I explain this? When you look at, do I have a structure? Did I put a structure in here?
I think whenever you show the two by each other, the eight,
I have a structure?
yeah, it was like eight, two different, yeah. I don't know if
But no, I'm looking for, there's a structure. If you go back in chapter,
I think FMN has the same, like the similar structure or the same.
have similar, it does have similar. So watch. So when you're picking up the electrons, you pick up the electrons on the carbon that are attached to the nitrogens that bond to the hydrogen, right? And so for because you're in a ring structure, right? When you pick up that one hydrogen, it only picks up one electron. So you have to pick up 2 hydrogens in order to pick up 2 electrons for FMN and FAD.
Okay, so it's just a different process because of the ring, basically.
Because of a different structure, yes.
Okay. Thank you.
Yeah. Anybody else?
So we didn't really get to the Q cycle.
We didn't really get to the Q. I thought we were getting to, it's because we spent so long talking in the beginning about finals.
Because I was trying to think about it.
No, we didn't. We didn't get to the Q cycle today. All right, I'm gonna go ahead and end the Zoom. If y'all have any questions, let me know. Y'all have a good rest of your Wednesday, and go do the review thing, whatever that is.
Self check.
No, the
Oh,
evaluation, course evaluation thing.
Oh, yeah.
evaluation. All right. Oh, I did that almost a month ago.
Oh, good.